So good morning again. My name is Lauren Close. I'm the Operations and Communications Manager for our team here at the Northeast Big Data Innovation Hub at Columbia University, and I'm also a member of the COVID Information Commons project team. I'd like to introduce you to the COVID Information Commons, or the KIC. We are a COVID-19 research collaboration platform brought to you by the Big Data Innovation Hubs and funded by the National Science Foundation's Convergence Accelerator. The KIC brings together scholars from all over the country and around the world to share their research findings in the form of lightning talks. And today, each of our speakers will engage this community directly, answering your questions about their work during a Q&A session at the end of our presentations. And I should begin by noting that we, of course, at the COVID Information Commons are committed to providing a supportive and welcoming environment to everyone who works, studies, and interacts with us at the Northeast Hub. We encourage you to review the below code of conduct for our online programs, and this includes information about reporting and accessibility features. I can share a link to those resources in the chat as well. And this afternoon, I want to also introduce you to two other members of our team that are on the call. We have Florence Hudson, our Executive Director, and Emily Rothenberg, who's our Program Manager for the National Student Data Corps. I'm now going to pass the virtual mic over to Florence, who will be sharing some background information about the KIC and our programming.
Thank you so much, Lauren, and thank you for always organizing these. You do such a great job. We really appreciate it. So thank you, everyone, for joining us today. Thank you to our speakers, as well as to all of our participants, whether you're on the live stream or you're in the Zoom with us, or if you're watching this on YouTube afterward. Share it with all your friends and colleagues so that everybody can benefit from all this great research being funded by the U.S. National Science Foundation, National Institutes of Health, and CDC. So as you heard, my name is Florence Hudson. I'm Executive Director of the Northeast Big Data Innovation Hub, which is headquartered at Columbia University in the city of New York. There are other Columbia universities, so that's why we always say that. And I'm the PI, or Principal Investigator, on the COVID Information Commons. A little background on the KIC. The National Science Foundation contacted us in March of 2020 when everything started shutting down, and we realized we were in a very big issue that we had to deal with. And they created this rapid award program. I call it rapid in, rapid out. They, they ask for proposals, they make decisions quickly, they get the money out so we can try to address this compelling issue for our country and actually for the world. So they contacted us in March of 2020. We went, they said, could you create a portal for people to easily find government-funded research, NSF-funded research related to COVID? We said, sure. So we went into the NSF website that day. I'll never forget this, there were exactly 32 awards related to COVID that day. Now we have over 13,000 in the COVID Information Commons, the good news and the bad news. So we were meant to create a portal. In our kickoff meeting, we had a webinar like this in July of 2020. We actually had like 178 people that joined us. And we said, well, rather than it being just a portal, welcome to the KIC community. We had 2 researchers present their research so that it wasn't just dry like portal stuff, like here's a portal, put your award number in, put the name of a human or an institution. And the 2 researchers did a great job. And then 40 other researchers said, we want to present as well to other people. We want to enable collaboration and get our research and information out there, find other research assets or data sets we could collaborate with. And so the KIC community was born. And so since then it's grown. So as you can see, it serves as an open resource to explore research addressing the COVID-19 pandemic. You could look for the COVID awards and PI database. You can go in there. There are over 13,000, as we said. You could put NSF, NIH, you could be an institute or a directorate. You could put a human name, an institutional name. And we also have this really cool machine learning maps tool. And so the initial award was in 2020. And then in 2021, we were so grateful for this funding, but this is a disease we really feel like we should work more closely with NIH as well. And so NSF gave us $2 million more in 2021 to work on this for 4 more years. And that was the KIC Extension Award. And so since then, we have included the NIH awards. We have a system architect that does an API scrape once a month and pulls all the new awards in. And as compared to, just it being about, the coronavirus itself, it's the implications of COVID, education, society, mental health, all the things that, have been impacted by COVID. So this continues to be a vital resource. It's really grown a lot with Lauren's help in creating a student working group and researcher working groups where they learn how to use data science techniques to look at information, data and metadata related to COVID. And so it really is a dynamic community that we're very grateful that all of you are participating in.
Yes, and I also, Florence, wanted to share these two other views of the Extension Award. This is the database as updated. Like you mentioned, we're just underneath scraping the bottom of 14,000 awards at this point. And I also want to share this view, which is, I think, much more in your wheelhouse than in mine in terms of this deep analysis.
Yeah, my favorite little thing. I feel like it looks like a little kaleidoscope or like, a window in a church or something, or a temple or something. So this is very interesting. This is actually a tool, Lingo4G, that NSF was using to create a map like this on the NSF website at looking at their different awards. And we were like, ooh, this is really cool, So we actually brought it into the COVID Info Commons. And we work with this little company in Poland that has this tool, and they do a machine learning clustering of the research awards. And so it shows all of them, or this is very hard to see, like, there are 13,000 of these on one little screen. So you can actually do queries like epidemiology or mental health or California or Florida or Louisiana or whatever you want to look at. And then the awards that mention that, it could be a school in Louisiana or it could be a study that was done in Louisiana, because, it kind of goes by the keywords. And then you can look at that, and then it categorizes them, as you could see. And you could look and say, oh, wow, I'm very interested in dynamics, transmission, and the epidemiological impact and view. And so who is doing work in there? And then you'll see, oh, wow, there's someone at Columbia University. I wonder who that is, when you can click on that, and then you can see the research information, the PI, the institution, how much money they got, when they were awarded it. A lot of this research is longitudinal, as people kind of keep on going. So it's really a great opportunity to find other research and other researchers. So these tools are out there, open, online, and free. And so we encourage you to share this with everybody, and if you have students that you work with, encourage them to learn more about the KIC student working group that Lauren's probably going to talk about too.
A perfect transition, Florence. I am actually, in fact, just about to talk about the KIC student working group. I do definitely want to plug this opportunity through the COVID Info Commons. The student working group is a relatively new initiative that we have from our team. We currently, however, have over 1,400 student members who are from all over the world, and this is a really great international collaboration space for STEM students who are interested in working together on data science projects related to all different areas of COVID. We've just wrapped up a couple of these different student projects that you can see here on the screen, the most recent of which is the Data Essentials project. So we definitely encourage you to pass those over to your students. We think that they will learn a lot about data and about COVID research through these initiatives. And our next meeting will actually be at the end of January, Friday, January 24th, when we will be launching a new data science project for the spring semester. These projects and programs are open to everyone free of charge, so please do consider joining. The other item that I want to mention here is just, again, this Data Essentials project that we launched in September. Students can join this project at any time. Everyone who participates gets a bit of a basis of data science analysis in Excel. It's a great project for students who want to learn those core fundamentals of scientific analysis from the ground up. It's also really great for students who are graduating and looking for jobs in data science. We've heard from several students in our community that recruiters are actually looking for Excel as a new desirable skill. Surprising to us, but a great opportunity for us to build a useful tool for them all. And we, through the KIC, provide a certificate of completion for each submitted project. Final projects may be shared on our website. So again, I encourage any faculty who are with us today to take a look at this project. We've also helped a couple of faculty at other universities, like the University of Washington, Tacoma, adopt this to their STEM curriculum. So it's useful for students as well as you who are on the faculty end of things. And I will also give one final plug to our other data science projects through the COVID Information Commons Student Working Group. But without further ado, I want to now introduce the speakers of today's COVID-19 webinar, because this afternoon we will be hearing from 4 speakers. It's a range of CDC and NIH-funded projects. And I would say to our audience, if you have any questions for our speakers during the presentations, those of you who are joining us through Zoom, you can feel free to drop them into the chat, or you can hang on to those questions for our Q&A session. Those of you who are joining us through our live stream, you can also drop those questions to us directly through the YouTube live stream. And I will stop sharing my screen and stop talking, because I'm going to actually pass over the virtual mic to our first speaker for today, who is Dr. Harvey Pollard. And Dr. Pollard, here are his slides, is joining us today from the Uniformed Services University School of Medicine, and he'll be sharing with us his work on respiratory inflammation and spike proteins. So Dr. Pollard, it's all yours.
Thank you. So good morning, everybody, or good afternoon. The title of this talk is Inflammation in the COVID-19 Airway is due to CFTR signaling by the SARS-CoV spike protein. Since we've all been living through the COVID pandemic, a lot of this will not be new to you, but CFTR may be new. And it stands for, it's the name of a protein, mutation which causes a disease called cystic fibrosis. This is something we work on. It's hard to remember actually the name of this protein, because the people who were studying it in the beginning at the Hospital for Sick Kids in Toronto had no idea what it did. But they were listening to a rock station, CFTR, Canadian Frequency Toronto, 680 on the AM dial, and they decided to call it CFTR. Actually, that's the way I remember it. And it's a way of sort of giving information about what people were doing in the old days when they didn't know what they were exactly doing. But in any event, the protein itself is really critical, as are many of the proteins or genes that are found in rare diseases, because they're rare because it's difficult to live without them. We are joined by colleagues at USAMRIID, the United States Army Medical Research Institute of Infectious Diseases, where the very worst viruses are actually studied. And we were able to, so this is a collaboration. And the paper that much of the data I'm going to talk about was recently published in Scientific Reports. Hung Kawi et al. Down below at the bottom. If we can go to the next slide. So the goals of this project were to understand, first of all, the mechanism by which pro-inflammatory cytokine storm in the COVID-19 airway occurred. It's essentially lethal. And second, to find a therapeutic strategy to suppress the storm. And by the way, if you see the spike trimer moving, it's supposed to be moving. It's not your eyes. Next slide. So firstly, we developed an in vitro assay for spike protein binding to ACE2. And then we looked for candidate drugs that could block the interaction. And finally, we found that cardiac glycosides such as digitoxin, digoxin, or wabane, these are all cardiac glycosides, were potent inhibitors of spike ACE2 binding. So after going through a lot of studies that were looking at the binding, we asked the question whether any of those drugs, which are actually approved around the world and licensed, digoxin is licensed here in the United States, actually interfered with infection. So on the next slide, this is basically a slide that shows that the infectivity of native virus is inhibited by digitoxin and other cardiac glycosides in human lung cells, the A549 cell, very typical. So across the top is a series, well, this is a series of dose response curves. The x-axis concentration of drug, the y-axis in green, inhibitory values, K1.5 values, essentially EC50 values, for all three of the cardiac glycoside drugs, and they're in concentration ranges used to treat heart failure, interestingly. The red colors is cytotoxicity. It's about tenfold greater than the respective inhibitory EC50 values, which means that you can use these drugs without getting into inhibiting the cells. The two boxes below the genins are the steroid nucleuses that go with these drugs minus the sugars, and they're not very effective. And that was published in Scientific Reports in 2021. So we were very happy to see that we could actually transmit some in vitro experiment into something that was approximating in vivo. Next slide, please. So after giving you, there's additional preliminary information about CFTR, which I want to emphasize. It's a gene responsible for the control of inflammation in the rare genetic disease, cystic fibrosis. And as we will see down in the next few slides, essential for control of inflammation in the COVID-19 airway. Next slide. So I'm just going to read the first part because it says what I want to say, and then we'll go on. Early in the COVID-19 pandemic, it was found that the lethal cytokine storm was due to activation of the TNF-alpha NF-kappa B pathway. But how that happened, no one knew. In the middle of this slide, you can see a signaling pathway which starts with, at the top, TNF-alpha, which binds then to a TNF receptor in the membrane of the cell. And there it actually connects up to the first intracellular adapter, a molecule called TRAD. The DD stands for death domain, very unnerving. That molecule is a critical director to take all those signals from TNF-alpha to the IKK-some that then binds to or phosphorylates I-kappa B alpha, releases NF-kappa B, which moves into the nucleus and generates all of these cytokines, IL-6, IL-8, TNF-alpha, and a bunch of, as messenger RNAs, which are then synthesized and secreted into the airway and into the rest of the body, actually, in the context of a cytokine storm. In studying this situation with cystic fibrosis, we found that there were some, looking at countervailing candidate drugs like digitoxin. We've already shown you that they interfere, by the way, with the infection. We found that digoxin, Wabane, basically block that process. You can see the little in red letters and little block, a little T-junction, showing that when you make a binary complex between TNF-alpha and TNF receptor 1 and it wants to bind TRAD, it can't. So that is, the first experiments were just done in plain old HeLa cells, and that was actually demonstrated where the problem lay. One function of CFTR is to block that molecule, TRAD, and thereby inhibit the cytokine storm in the CF airway. But in fact, since the digitoxin and the CFTR are all working in the same place, essentially the drugs are phenocopying or mimicking the CFTR, we hypothesized that the COVID-19 cytokine storm was due to loss of CFTR, and that cardiac glycosides, which would basically mimic CFTR, might be therapeutic. Next slide. So the first question was, does spike protein exposure reduce CFTR? The way we actually did this experiment, we used a human organoid lung preparation, prepared at the air-water interface. You put some basal cells, which we got from a colleague, into this air-water interface. And over the course of 28 days of farming, you grow a beautiful epithelium. You can see goblet cells and ciliary cells and other sorts of cells verified by immunofluorescence below. So on the right-hand side, we are using Western blot analysis of spike protein effects on levels of CFTR. Along the horizontal axis, we're increasing the concentration in the first upper column, A, ancestral or original, the Wuhan version of the spike protein, and it causes a significant dose-dependent loss of CFTR. We then looked at a much more biologically potent strain. This is the South African beta strain, much more potent. It turned out as a virus to cause COVID, but much more potent at reducing CFTR. You can see the difference in the bar graphs and in the Western blots. So yes, we concluded spike protein exposure does reduce CFTR levels. Next slide, please. Next question was, does spike protein activate pro-inflammatory NF-kappa B signaling? Well, in the picture that I showed of the signaling pathways, there's a whole series of proteins starting from the top, TNF-alpha and TRED and so forth, which get activated if CFTR is lost. Well, as you increase the concentration of the spike protein, again, we're using the Wuhan level, the strain here, all of those red arrows on the left actually show places where increases is happening. And if you look over, you'll see, yes, TRED is actually increasing. And in the end, IL-8, which is one of the cytokines we measured, is also increasing. So yes, the spike protein activates pro-inflammatory NF-kappa B signaling. Next slide. Question at that point, and this is where our USAMRIID colleagues came in handy, do the cardiac glycosides protect lung epithelial cells from CFTR loss by native virus? On the left-hand side, you'll see a Western blot. Various things are added. Right in the middle, there where it says virus only, you'll see that the level of intensity of that CFTR band, in this case it's the dimer, is reduced substantially compared with the medium control. And that digitoxin and wabane raise the levels. A bar graph basically shows very similar things. These are all based on 4 independent experiments. And the real situation here is that it's all very well for us to actually do experiments with spike proteins, but it's very important to verify that this is happening in real viruses and real disease. So this was something that we really were interested to show and to validate. We'll go on to the next slide, please. So the question was, how does spike which we know only bind to ACE2, yet it also seems to affect CFTR levels? How could that be? One possibility was that maybe the CFTR existed bound to ACE2 in the membrane. Actually, CFTR is bound to a lot of things in the membrane. Well, it turns out, yes, it's bound to ACE2. The left-hand side of this figure shows that, in fact, CFTR and ACE2 actually are found both in the basal cells that we make the differentiated epithelium from, and in the differentiated epithelium. CFTR is there, ACE2, and actually everything else you need to actually get a nice infection going, as long as you can call an infection nice. On the right-hand side, it shows that if you take an antibody against ACE2 and immunoprecipitate it, and then test that immunoprecipitate for bound CFTR, sure enough, you can find plenty of CFTR. Those are 2 duplicate experiments. The NRS is just simply normal rat serum. You can also find immunoprecipitated CFTR in just the basal cells. So it's basically accessible in all the different parts of the epithelium. And then, next slide. I want to pause here to remind or to emphasize to everyone, CFTR is a cyclic AMP activated chloride channel. That channel is critical for keeping the airway hydrated. When the channel isn't there, the airway dries out and inflammation occurs. But in this particular case, here we have the CFTR protein in the cell surface. Up is airway, down is the interior of the cell. Norepinephrine or epinephrine can bind to a protein that actually does bind another protein that binds the CFTR called the beta-2 adrenergic receptor. Generates cyclic AMP, protein kinase A is activated, and it then labels the phosphorylates the CFTR, and the channel activity is activated. You can see a chloride molecule moving out. Why do I show this? Well, if the protein is being lost, you expect that the channel activity would also be lost. It's sort of a test. Is it really true that the spike protein does remove everything associated with the CFTR? So let's look at the next slide. And on the right-hand side is a classical Using chamber experiment, essentially. It's what they call it. But we could actually look over the horizontal axis on the left-hand side is time course, and the vertical axis is the activity of transport of chloride, actually from one side of the air-liquid interface to the air side. And if you add some IBMX or something to stabilize the cyclic AMP, the green, the channel actually opens up, and you can see a very nice signal. And if you add an inhibitor of CFTR, it will go down. But if you add spike protein, then you lose about 50% of the signal in this experiment. Spike in this case is Wuhan, so it's even the least potent of these. But if you add Wobbing or digitoxin or digoxin to this cell culture, it's culturing over the course of 24 hours, of course, the spike effect is lost. On the right-hand side, you can see that story with the bar graphs, but just below it, where it says CFTR, you can see that when you've added spike, you even get less CFTR. So the spike protein causes loss of the CFTR protein in the same system, and the CFTR protein loss is rescued by treatment with of the epithelia with all three cardiac glycosides. So the loss of activity, channel activity, loss of protein, all occur at the same time. That means we feel we've robustly addressed that concept that the spike protein or the virus, for that matter, has reduced CFTR levels. Next slide. So our conclusions are that the data support the hypothesis that the COVID-19 cytokine storm is due to loss of CFTR. Second, the loss of CFTR activates TRAD to drive TNF-alpha, NF-kappa B signaling. Digitoxin and digoxin and Wobbing may be therapeutic by blocking, first of all, by blocking infection, by inhibiting the virus from binding to ACE2, and second, by inhibiting inflammation, by inhibiting TRAD, and thus substituting for the lost CFTR. We suggest that the cardiac glycoside advantage is that it is one family of drugs with two simultaneous candidate therapeutic activities for COVID-19. Next slide. We have to have these acknowledgements, partly our lab at Uniform Services, and also the Herbert and Flores lab at USAMRIID. And we also have to have a Department of Defense disclaimer, which talks about the fact that we're not trying to sell anybody anything. And we thank you for that. Next slide. Yes, thank you. And I look forward to any questions that might be there later in the presentation.
Thank you so much, Dr. Pollard. I want to say to the group that if anyone has any questions for our speakers, whether you're on the live stream or in the Zoom with us, again, please feel free to drop those into the chat, or you can hang on to them for later in our webinar session when we will host an open Q&A. But now I want to hand it over to our next speaker, who is Dr. Emily Wiemers at Syracuse University, and she'll be talking about her team's research on family health during COVID. So Dr. Wiemers, the floor is yours.
Thank you so much. So we're really going to shift gears here and think about some of the secondary effects of the pandemic and how families helped one another during that. So this is research from part of a larger project that looks at the family support during the pandemic, and it's joint work with folks at Syracuse, at Bowling Green State University, at UCLA, and at the Harris School at Chicago. So just to give everyone a little bit of context, families are incredibly important in terms of caring for older adults. So the care that family members give to adults with activity limitations is worth about $600 billion a year. And adult children are a big part of this system of family care. They're about half of the primary caregivers for community dwelling older adults who receive care. And while kids are very good about giving their parents time help, they also give them money. So in general, parents tend to give their kids money for most of their lives. But when parents get old, and particularly when they have declining health or when they face sort of economic distress, there is evidence that adult children do step in and also provide their parents with money help. So when we think about this in the context of the pandemic increased the need for help among older adults and among everyone, right? So while actually the economic impact of the pandemic on older adults was actually slightly smaller than it was for younger adults, it was longer lasting. And also older adults faced additional challenges, and in particular, they faced logistical obstacles for things like getting food, partially because of the safety of going to the grocery store, for example, but also because things like using apps and things to do grocery delivery were more difficult for older adults. And as a consequence of this, during the first few months of the pandemic, food insecurity increased a lot among older adults. It increased by 75% compared to pre-pandemic levels. And usually when these bad things happen to older adults, their adult children step in to help them. But as you know, especially during the first year of the pandemic, bad things were happening to everyone, right? And so while older adults were more needy, their kids were also more needy. Their adult children were also more needy. So on the one hand, we know that adult children did step in to sort of provide unpaid care when paid care became very difficult to obtain for older adults who had lots of health difficulties. So, for example, older adults moved in with their children to avoid going to nursing homes and things like that. But on the other hand, middle-aged adults had lots of challenges, including the fact that many of their kids weren't in school, which may have limited their ability to provide sort of care that was not like, absolutely medically necessary. And also, if you remember back to that time, we were very nervous about infecting our grandmothers and parents with the illness. And so we, in some sense, tried to maintain physical distancing wherever possible. And so, it could be that it was difficult for kids to provide the things they would normally do because of the need for physical distancing. So this project or this paper that I'm talking about looks at how responsive kids were with money and time help during the pandemic, and whether they were responsive to specific needs. That is, did they give money when their parents needed money, and did they give time when their parents needed time? And we've also been interested in whether kids were more responsive to their parents' needs during the pandemic than they had been in the past. And one reason this is interesting is to try to think about whether this is something that going forward we might expect to see more of, or whether this was just part of like what kids always do. Okay, so we're going to use data from the Health and Retirement Study to answer this question. The Health and Retirement Study is a nationally representative longitudinal study of older adults. It's been ongoing since 1992 and is sort of the gold standard for studying the health and well-being of older adults in the United States. In 2020, they conducted a special module for respondents who were interviewed after May of 2020, in which people were asked about transfers of time and money that were given to and received from others outside of their household because of the pandemic. And I'll talk to you a little bit about what they were specifically asked in a little bit. But this is also a study that's longitudinal, so it's been going on every year. And the people who were interviewed in 2020 were also, most of them were also in the study in 2016 and 2018. And so we also use information from the past to ask this question about whether kids were more responsive now than they had been in the past. And the regular core, this comes from the regular core interview, which includes lots of demographic, economic, family, and health characteristics, as well as questions about time and financial help received from adult children in the context of needing help with activities of daily living or instrumental activities of daily living, including help providing and preparing food. So it's really hard to figure out exactly how to nail down exactly what caused what during the pandemic, because if you remember, everything was happening at once, and what caused what is a big chain that's difficult to unravel. So we kind of use this like complementary strategies where we look at different ways of looking at this question and see if we get the same answer when we look at it several different ways, and then use that to kind of triangulate a conclusion. So the two ways I'm going to talk about today is, first, we just use what parents say they needed and what they got from their kids during the pandemic. And then we also look at what happened in the past. So we're going to look at change over time from 2016 to 2020 as a way to answer this question about whether kids became more responsive during the pandemic than they had been in the past. And we use a sample of about 6,000 older adults who participated in the 2020 COVID-19 module, but were also interviewed in the 16, 18, and 20 core. We use people who are age 55 and older because the cohort aged, and so we want to keep the age group consistent. And we include people who have at least one non-co-resident child because the questions that were asked were about help from people outside your household. So in terms of what they were asked in the COVID module, they were asked, because of the pandemic, did anyone living outside your household, such as a parent, adult, child, other relative, or friend, help you with money or by paying bills? And if so, who was that? And did they help you with shopping for groceries, errands, rides, or chores? And if so, who was that? People were also asked about challenges that they faced. So they were asked if they missed any regular payments, like on their rent or mortgage or other bills, whether they couldn't pay medical bills or whether they didn't have enough money to buy food. And we consider someone to have experienced economic hardship if they report any of those. And about 13% of the sample did report that they had economic hardship. Respondents were also asked if they had trouble buying food even though they had money. And so we're going to think about that as them having trouble buying food for non-financial reasons. And about a similar percentage, about 14% of the sample reported that they had trouble buying food for non-financial reasons. In the core survey, they're asked slightly different things. So the concepts are the same, but the exact measurement is a little bit different, which actually I think helps us in terms of this triangulation approach, because we don't want our conclusions to be based on exactly certain question wording or types of help that was received. So in the core survey, people are asked if they received money from their children and if they received $500 or more over the past 2 years. They're also asked if they received time help for things like meal preparation and shopping for groceries. So that's our measure of time help from the core. In the core survey, they're also asked about need. So we code someone as experiencing hardship if they report missing rent or mortgage payments or not having enough money to buy food. And we record them as having trouble buying food for non-financial reasons if they have difficulty shopping for groceries because of health or memory problems. So you can see that in that's sort of linked to a health problem, whereas in the COVID-19 module, it's not linked to that health problem. The survey, of course, is very big and gives us lots and lots of controls that I'm not going to talk to, but what I'm going to show you is all net of those controls. And because we have people interviewed over time, we're able to use prior waves information about your income and health so that we're not looking at things that are happening at the same time. We're controlling for your prior circumstances. So I'll show you a little bit of the results. So here, this shows the coefficients on models that predict money and time help that older people receive from their adult children. So what these show on the left-hand side is that when parents reported that they had economic hardship, they were 6.4 percentage points more likely to report that they received money from their kids. But if they reported economic hardship, they weren't more likely to receive time help from their kids. That's that 0.1%, right? Similarly, if they reported that they had trouble buying food for non-financial reasons, they were 7.7 percentage points more likely to say that a child helped them. But if they reported that they had trouble buying food, they weren't more likely to get money help from their kids. So this shows us that kids were responsive to the needs their parents have. That is, they gave money for money problems and time for time problems, right? But that doesn't really say anything about whether this was changed over time. So here what we're looking at is we're looking at that same thing. We're looking at how having hardship affects the probability that you get money from your kid. And we're looking at that in 3 different waves, in 2016, 2018, and 2020. And similarly for food, we're looking at how having trouble buying food for non-financial reasons affects the probability that you get money from your kids in 16, 18, and 20. And what we can see here on the left is that only in 2020 does having economic hardship predict getting money from your kids. If in earlier waves, having economic hardship does not increase the probability that you get money from your kids. So here we can conclude that kids were more responsive to financial needs during the pandemic than they had been in the past. But in none of the waves did they respond to having trouble buying food by giving their parents money. In time help, we have something similar. So on the left-hand side, we're looking at whether having economic hardship predicts your kids giving you time help. And you can basically see in all of the years, there's very small associations between having economic hardship and getting time help from your kids. And you can see on the right-hand side that in every wave, having trouble buying food for non-financial reasons increases the probability that you get help from your kids. But what you can see if you compare 2016, 2018, and 2020 is that in 2020, experiencing trouble buying food for non-financial reasons increased the probability that you get time help from your kids by 20 percentage points, compared to only about 10 percentage points in earlier waves. And so this shows that kids were more responsive to their parents' time needs in 2020 than they had been in the past. So just to summarize, what we find is that adult children were quite responsive to their parents' specific pandemic-related needs. That is, they gave parents money when they needed money, and they gave parents time help when they needed help for non-financial reasons. And we also showed that adult children were more responsive to their parents' needs during the pandemic than they had been in the past. And I think that runs contrary to a lot of the sort of concerns during the pandemic, which is that older adults would sort of be abandoned and alone. In fact, we find that their kids were more responsive. There are lots of limitations. We're just looking at the first year, and we're relying on parents' reports, but this is like a gold standard survey. And so we feel like these are pretty robust findings. I'll just acknowledge that this was funded by NIA, and the grant number is there. We're very grateful to the funding, and this has been published recently in the Journal of Marriage and Family, and the citation is below. So thank you very much. I appreciate your time.
Thank you so much, Dr. Wiemers. You're right. That was quite a switch. And I think we're going to maybe make another pivot, another switch, because I'm going to introduce us to our third speaker this afternoon, Dr. Gabriella Wilson, who is coming to us from the University of Louisiana, Lafayette.
Hello, everyone. Can you hear me? Well, thank you so much. I was looking forward to this gathering. The first time when I was approached about participating in this panel, I was at the University of Texas at Arlington, and I was transitioning to University of Louisiana Lafayette. And I have now 4 months in this job, doing lots of things that are somehow related to what I did in the past, but because of the project on COVID-19, the focus of my presentation is going to continue to be on the work that was funded through the CDC with the University of Texas at Arlington. So I will describe, and for some reason I cannot advance my slides.
Try the right arrow. I had a problem with the down arrow before.
Hold on one
Okay.
second. I think I got it. Okay. Can you see?
Yes, great.
Okay, wonderful, wonderful. So the title of the grant that we received through the CDC and mainly through the Tarrant County Public Health Department, where University of Texas at Arlington was located, was National Initiative to Address COVID-19 Health Disparities Among Populations at High Risk and underserved, including racial and ethnic minority population and rural communities. So the entire Department of Public Health in Tarrant County received $16 million. And they were really good connecting all community partners to be part of this big initiative. So the intended outcome of this funding was really to reduce COVID-19 related health disparities, and there were multiple public health departments across the country that received funding. Texas was able, Tarrant County in particular, was able to have a very compelling story because we had Hispanic population now is majority minority in North Texas. And we could see that there was reluctance in accepting vaccines, or there were some stigma and lots of misinformation. So part of our focus was really looking at minorities, but with a focus on the Hispanic population in North Texas. So what we wanted to accomplish through this grant was to improve and increase testing and contact tracing at that time. So this funding was for a period of 3 years among the populations at high risk, those underserved, and with a focus also on the rural communities. And let's face it, Texas has lots of rural communities, especially North Texas. The goal at the national level was, and in particular here, Texas, was to improve state, local, and U.S. territorial and freely associated state health department capacity and provide more services in order to be able to control infection among populations at high risk. So part of the approach that we used in Tarrant County was to focus on 3 areas. So it's like a stool with 3 legs. We want to reduce disparities related to COVID-19, but how do we do that? So it was clear from the beginning that we had to have a very strong communication messaging initiative across the North Texas, all be consistent messages that are being disseminated across different groups. We also needed to involve community in the process. So, community health workers, public library, faith-based organization, grassroots, etc. And then also we needed to focus on countering the misinformation that was going through different areas that would play a role in the marketing, in promoting the vaccine and all the initiatives that the public health department was trying to promote. The second leg of the stool was partnership was going to be extremely important. So train with and include more partners to do the contract tracing, include transportation so people can go to different pharmacies, for example, where the vaccine was going to be distributed, have mobile units in place in the areas where there were no pharmacies that were delivering the vaccine, make sure that everybody has access to distribution of the vaccine, and of course, other barriers such as PPE, limited paid leave, and etc. And last but not least was to focus on reducing the cultural barriers. So make sure that we use the trusted resources, connections for vaccine sites, address the language and cultural barriers that existed. So I mentioned we are heavily in the Hispanic population, and we wanted to make sure that COVID-19 is not being translated as the gripa, which means influenza, in fact, in Spanish. So we noticed there were lots of announcements at the airport, for example, where the translation for get tested for COVID-19 were translated get tested for gripa. And in the Hispanic population, gripa is not COVID-19. Gripa was really influenza, gripa. So that was something that we wanted to address, so people do not think that this is just a gripa, an influenza. It's something much more complicated. Build and improve the relationships with the communities, especially those underserved, and of course, involve the community in the entire process from the beginning until the end. So as a PI for the UTA side, I wanted from the beginning, and again, I'm a health informatician, moved into public health informatics during the pandemic. At that time, I was still in Indiana, moving into a new role at the University of Texas at Arlington, and I started tracking the number of COVID cases, first in Indiana, and then I said, wait a minute, I am starting a new job. So I started my job in Texas, March 1st of 2020. March 13 was the pandemic, when the pandemic was declared. And I couldn't move to Texas until August. So I did all this work first, tracking and making sure that I understand the community where I'm going to move. And then I realized this is much bigger than just me as an individual tracking cases, but it's actually something that I think that we can help with data, analyzing the data, identifying the high-risk neighborhoods, and all of these aspects. So I wanted from the beginning, as a co-founding co-director of a center focused on health informatics, to establish the center as a good partner for the university, but also be part of the solution by collaborating with community partners to prepare and monitor cases COVID-19 in Tarrant County. And lots of this work was done for free at the beginning, just because we care about the community. And then, of course, by connecting with Tarrant County Public Health, I got the opportunity to also receive funding. So from the beginning, the way that we approached this as a university, my entire team was, we wanted to build trust in our citizens to accept the COVID-19 vaccine when that would be available. At that time, it wasn't, we were just talking about it. We participated in contact tracing and other things, but we knew when the vaccine is going to be available, we wanted to make sure everybody has access. Then we wanted to provide to the Tarrant County communities in high-risk neighborhoods information that is culturally sensitive related to the vaccine and COVID-19 health in general. We wanted to also be able to support the equitable distribution in high-risk neighborhoods. And we wanted to be able to continue to monitor the impacts of the pandemic, because at that time we were thinking it was going to be just a few months or maybe 1 year, but we all know that it took longer than that. And that's why it was very important to monitor, continue to monitor the impacts of the pandemic, vaccination, emerging variants, and even now long COVID. People are still working in this to see the impacts on general health. So I'm going to describe a little bit on how we supported equitable COVID-19 vaccine distribution in high-risk neighborhoods, and how we used the data and health informatics and public health informatics to address these needs to support public health department in Tarrant County. So what we thought about was people, they need to see it on a map to have a better visual instead of just graphs and data and correlations and coefficients. In public health, as you probably know, it is much easier when they look at pinpoint, okay, the zip code level, the census tract level, where is this located? So for us, the focus was from the beginning to do a data-driven health intelligence approach in preparing for vaccine distribution and monitoring of the pandemic. So what you see in this map is Tarrant County, where we are able to identify high-risk neighborhoods at the census tract level. So what you see darker red are those high-risk neighborhoods. We also we have a paper that I'm citing here that describes the entire approach. But by doing this, what we are able to show Tarrant County Public Health Department was where you are going to distribute the vaccine, where you see the pharmacies with the white cross, you see the high-risk neighborhoods that are showed in red here. They don't have a pharmacy. They didn't have a pharmacy in those areas. So we were able to help deploy the vaccination, the mobile units that will go into those neighborhoods to really provide information. The vaccine is available, but also do the vaccination. So that was extremely important, because what we did is not just adding a social vulnerability index. We also added all the SDOH data, including health literacy, and of course the demographics. But the health literacy played an important role here, even though the data was from 2017. We use that as an indicator. Where do we need to go and discuss more or provide more information in any type of form? It can be focus groups, where we did surveys on paper. We did sessions with church leader, faith-based organizations, with the public libraries, just to be able to provide the information that was needed in a very sensitive, culturally sensitive manner. So how then, for the next step, how did we monitor for the impacts of the pandemic and the vaccination rate? So that was a second part of the study that we did. And we kind of focused our research on two hypotheses. We all love social media, right? So we also know that when the vaccine became available, everybody was super excited and everybody wanted to get the vaccine, even though it was age categories. So first we vaccinate the elderly and those who are more vulnerable. Then it was the teachers. I'm not talking about, of course, the healthcare workers who are frontline and they received the vaccine first. But lots of people wanted to get a vaccine even if it wasn't the time. And there were huge lines, I remember, in auditoriums, in stadiums across North Texas. And then all of a sudden, people started talking about, you shouldn't get the vaccine, you shouldn't do that. So what we did, we wanted to use social media to be able to analyze the sentiments that can influence vaccination rate. Is there any correlation? And then what other factors contribute to vaccine hesitancy? So all of this was done, of course, in combination with interviews and focus groups in the community. But my group focused mainly on analyzing social media activity. We did that. First, we started with Twitter, now X, analyzing the data. And we realized that the Hispanic population was not using Twitter. They were using Facebook. And because of that, we said we have to study in both English and Spanish. So we developed, we use natural language processing, machine learning, AI, everything that was at our disposal to really be able to identify what sentiments are being shown by the population in both languages related to the vaccine. What is it that they worry about and they talk about? So then when we work together with our community partners in the public health department, then we can have the messaging done in such a way to address those concerns, instead of just saying, hey, you should take the vaccine because the government wants you to take it. So then we also look at the health literacy and social vulnerability index, and we realize that the vaccination rate is highly influenced also by the level of health literacy and, of course, the SVI index. So the conclusion of this study, and I listed here 2 of our publications. We added a few more after that on other topics. One is abortion, for example, and we know Texas was the first one for the pushing for the abortion law. So we wanted to also look at stance towards abortion. But social media is really a powerful way of really understanding how people feel about certain things. And instead of just saying that you shouldn't listen or go on social media to get information, we should all be very active. And instead of going into a bystander mode, and we have a paper on that too, when there is very toxic information being put on social media, those of us who know the truth and the science behind decide to remove ourselves from that situation instead of going on social media and providing the correct information. So all of these studies were correlated very closely with what we are seeing in the community through the interviews, through the focus groups, and it would, by education, we are able to, of course, increase vaccination rate in areas where it was much lower before. So we receive the data from Public Health Department on vaccination every month, and we are now in the process of publishing the study that correlates all of the social media sentiments with the vaccination rate, real vaccination rate from the Public Health Department. So the third piece of our study was also how can we help build trust and provide that cultural sensitive information that's so needed? And it was needed during the pandemic, and it's still needed now in many different aspects. So part of, even though it was not funded by the CDC, this was a different study that we wanted to include the high school students as our champions for providing the correct information to their families, with a focus again on the Hispanic population. And why? Because in Texas, you can become a certified community health worker at age 16. And we realized that some of these community health workers, certified community health workers, were in high schools. We identify high schools in the predominantly Latino neighborhoods where vaccination rate was very low. And we realized that if we are using these CHWs in high schools to train them on how to talk about COVID-19 vaccine, they will go home to their families. And we know the Hispanic families, they live in multi-generational households, meaning that whatever information comes from school needs to go to the parents and grandparents. They will discuss it and they will approve it or disapprove it. So if we have the right training of our CHWs in high school, they can go in those communities and really talk about the correct information that needs to be provided regarding COVID-19. Because of this project, so we developed a training for CHW in high schools in those areas of high risk. And we received because of the project, we received the inaugural Racial Justice and Equity Award by the Coalition of Urban Serving Institutions in 2021. And I am extremely proud of being able to get to that level. It really put on the map the efforts that UTA, University of Texas at Arlington, was doing in the community, knowing that UTA was, and it is, a Hispanic serving institution. So that was amazing for all of us. We are very proud. And we organized the first symposium on racial justice and equity at our university the following year because of this award. So that was great visibility. So there are way too many people that I should acknowledge here and thank. So what I thought was to really put in this diagram all the individuals, all the community partners that contributed to this entire effort. Of course, the University of Texas at Arlington, I worked with the libraries, we work with public health, with TESOL as well, all partnering schools, and of course the La Alianza in the DFW area with the community health workers as well, and Health Literacy Texas. So we all together, we are able to achieve much more than working in isolation. We are able to inform about COVID-19 and the vaccines. We are able to address the community concerns that existed. And I think we are able to really combat misinformation by having targeted messaging that was provided as a result of the social media analysis and the interviews we did in the community. So I wanted to end with this beautiful mural that was painted by UTA students during the lockdown, where nobody was able to go on campus. They will go one at a time, art students, and they would start building this beautiful mural that now reminds us. I hope to be able to go back there to revisit this, but really is a reminder of those times when we all came together and we wanted to solve a big problem that hopefully we are not going to experience again. So here are my references in case anybody is interested. Thank you so much, Florence. The two of us connected at the HIMSS conference of, I don't even know, last year. It feels like it's more than 1 year, but through this connection we made, I am so grateful and really proud that you included me in this beautiful panel. Thank you so much. I appreciate it.
Well, Gabriella, and also thank you for looking into your past when you were still at UT Arlington and bringing this forward. Hope you don't get in trouble with your new family in Louisiana.
Oh, no. We have so many great things. Hopefully next time we'll have a presentation about other things I'm doing here. So thank you so much. I appreciate it.
Great. Yeah. And I feel I'm very grateful that we met and that we've been supporting each other in our efforts. And I think we can continue to do that into the future, which really is what this KIC community is about, and the Northeast Big Data Hub community. The hub, actually, I joined as executive director in March of 2020 when everything hit the fan. And at the time, there were 1,400 individuals that had participated in hub efforts. And now we have over 13,700, which is absolutely amazing. And it's really because of our incredible team, Lauren and Emily and our part-time students. But in a way, this is terrible. It was the silver lining of COVID because we had to do everything virtually. We couldn't do anything on campus. So what that did is really increased our broader impact,
Yes. And I don't want to take more time, but Florence and I also collaborated on a NIH NLM grant that I have to train biomedical informatics and data science undergraduate students from underrepresented groups. And Florence, I want to tell you that I want to include you again because I was able to transfer that grant to University of Louisiana Lafayette. So I will not get in trouble, believe me.
That's excellent. The gifts that keep on giving. It's a beautiful thing. That's what I do too. All the projects I do, I tuck them under where I am. Right.
Thank you again.
Oh, our pleasure. And we look forward to that. And one of the things Gabrielle was talking about is that we did a presentation, had a meeting with her team, TEDx Bio, was it called, or something like that. And so we actually presented the National Student Data Corps, which is a sister program to the KIC. And we have open online free data science education. We have asynchronous data science projects that learners of any age can do. It doesn't care how old you are or what you know or what you don't know. You can go into these things. And so that's something that we're happy to, to teach other schools about. And we look forward to that, Gabrielle. It'll be a lot of fun. And I remember they were so collaborative. There were a lot of really...
Well, they created a chapter because of you at the university. So hopefully we do this in Louisiana too.
That's great. Then we can come visit for Mardi Gras. Oh, did I say that? Okay.
Absolutely.
Well, actually, I think the timing is is absolutely perfect because I believe Dr. Dumitriu, who is our fourth speaker for today, has just joined. Dr. Dumitriu, it's great to see you. Thank you for joining us this afternoon.
Hi, yes, thank you for having me. And sorry to just be popping in and out. I am in a conference and have a couple other things that are going on today. Can you hear me okay?
Yes, we absolutely can. And thank you again for making time to join us. We did not want to miss your presentation, so thank you for letting us impose on your schedule today.
All right, I guess can I share my screen?
You absolutely can. The floor is yours.
Perfect, thank you. So great to be here. Thank you for this invitation. Thank you for finding me. It's great to see the great work that is coming out of the KIC and really uniting us and trying to understand how these really uncertain times are going to affect our world. So I'm here to talk to you very briefly about our combo initiative, or COVID-19 Mother-Baby Outcomes Initiative, that we spearheaded at Columbia University. Lauren found me because we had one early grant, NIH grant, to study brain behavioral functioning, and I will tell you a little bit about this. But broadly speaking, as you'll see, this is a much, much bigger initiative than that. So I wear a lot of different hats at Columbia University, but one of them is, as a basic neuroscientist, I actually study the neural circuits of resilience, and that is all I did pretty much up until the pandemic. In my lab. And then the second one is I am one of the newborn hospitalists in our well baby nursery. So I'm not a neonatologist. I don't deal with sick babies. I deal with healthy babies 20% of my time. And before the pandemic, I used to say that this was my vacation time away from the lab where things are never working out. And I get to spend time with new parents and their adorable little babies. But on March 13, 2020, that all changed. So I'm not sure the composition of the room in terms of whether or not you're from New York or are familiar with the fact that we were the first epicenter, including for pregnant and laboring women. So March 13, 2020 is actually the date in our country that the very first woman that a woman in labor was first diagnosed with COVID-19. And so that baby was coming to our nursery, and the world was, of course, looking towards our nursery to come up with the protocols, the infection and prevention protocols, as well as just learning from our experience in terms of what to expect from these babies. And so as a neuroscientist and developmental neuroscientists in particular, one of the things that, of course, I immediately thought about is the concept of the developmental origins of health and disease, or DOHAD, also called prenatal programming, where we have really learned in the last couple of decades that virtually anything that happens to mom while she is pregnant can affect the fetus. So viruses and the consequent maternal immune activation are some of the insults, and there's lots of examples from the literature. Zika, for example, even in children that are not actually infected with the Zika virus, is associated with language deficits. A variety of viruses have been associated with psychotic disorders all the way into adulthood. And there's many other examples. We also know that things like natural experiments to any kind of stressor, like famine or natural disasters, war, also has been associated with the increased risk to the offspring, including schizophrenia, language and cognitive deficits, and difficult infant temperament. And of course, the COVID-19 pandemic was this perfect intersection of both of these really influential insults during pregnancy. As these children, as we were going into the summer of 2020, we also began to think about just being born into an altered world. So adverse childhood experiences have been shown to be dose-dependently involved in the increased risk of different diseases and disorders. Some of the things that you can think about very easily, like depression and anxiety, but also they increase the risk of things like cancer and even broken bones later in life. And so we very rapidly mobilized to start Combo. The very first dyad was actually enrolled on May 26, 2020. We began with simple phone calls that we physicians were making to our families that had been hospitalized in our well baby nursery just to check up on the baby. We rapidly began to add surveys. We also included a prenatal arm to enroll pregnant individuals. And then we've sort of been chasing this cohort ever since then, and we just began our 4 to 5 year assessment now. And as funding became available, we began to add various things like biospecimens, video visits, brain MRIs of both moms and babies, cool stuff like smell testing of the mothers. And we also have allowed entry point at every time point through the 24-month time point to really capture as big of a population as we can. We've been very fortunate to have early and sustained funding since basically spring of 2020 through both federal and non-federal funds. And most importantly, this is a very, very big initiative. This is actually the picture of combo from the summer of 2020, which contains 77 people. We are now well over that. So we've had about 200 contributing members to date. We've enrolled 2,000 mother-baby dyads. We were able to expand outside of New York into Utah and Alabama thanks to a CDC contract. And we just gather so much data that we don't know what to do with it. So if anybody here is interested in collaborating with us, we're completely open source and open science approach, minus the fact that I don't have the infrastructure to generate DTAs fast enough and be able to set up collaborations fast enough. But we are really interested in any kind of collaborative effort. We've generated over 20 publications at this point, and we've had many trainees that have used this data for their thesis, for their T32s and K99s. And we've generated some of the most foundational data on what is happening to the COVID-19 generation. This is coverage from an early paper that I'll talk to you a little bit about today. And what I'm going to do, because this is only a 10-minute talk, is I'm going to cover 4 papers in 1 minute each, just to kind of give you the highlights of some of the biggest findings that we've had. So this is our first paper that we put together in exactly 14 days in April of 2020 in order to look at the rate of vertical transmission. So if mom has COVID while she is pregnant and delivering, is the infant at higher risk of infection? So in this study, we looked at the electronic health records of the first 101 infants born to COVID positive moms at Columbia University. So these babies were born in March and April of 2020, which was that really acute peak of the first peak of the pandemic. And as I mentioned, there were 101 infants born during that time. That was a period of about 6 weeks that we looked at, the first 6 weeks of the pandemic. And the most important part about this research is that we were one of the only hospitals that did not separate moms and babies. And so we really felt that this was an important thing to look at very early on, both to protect our own patients, but also to contribute to the research necessary in order for the rest of the world to follow suit and stop the separation, which we know is very dangerous to separate moms and babies. And what we found was that there was a very low risk of vertical transmission, and the 2 babies that did test positive did not have any symptoms. And so this was work that contributed to changes in policy, including by the CDC and World Health Organization, and the ending of the mother-baby separation. I also have the QR codes for each of the 4 papers, forgot to mention. So if you're interested to see the rest of the manuscript, feel free to take a snapshot. The next paper that I'm going to tell you about came out in January of 2022. And this was the first paper worldwide really to look at the neurodevelopment of these infants. And for this paper, we used the ASQ3 or Ages and Stages questionnaire, which is widely used in both clinical settings as well as research settings, as online surveys filled out by mothers. This was specifically targeting 6-month-olds born between March of 2020 and December of 2020. And this was restricted to our population at around Columbia University. In this paper, we covered 317 infants, 141 that we knew had not been exposed to COVID in utero. And I'm not going to go through it, but if anybody has questions, we have some of the world's most characterized maternal infection for the year of 2020, thanks to both hospital policies that implemented very early universal testing, as well as research strategies. We had 114 infants that we knew to be exposed either in the first, second, or third trimester to maternal infection. And we also were very fortunate to be able to include 62 infants that had been born in our hospital system in the preceding 3 years before the pandemic. And to summarize the finding of this paper, we found no effects of COVID exposure, but we did see some small decrements in pandemic-born children. So this indicated to us that it might be the maternal stress or distress experienced during pregnancy because of the pandemic, rather than an actual viral infection effect. But of course, that was maternal report, and the gold standard in the field is to actually test infants with objective measures of development, with actual neurodevelopmental assessments performed by either clinical or research staff. And so in this second paper, we followed up using an observational tool called a DAISY-2. For those of you who might know a little bit about child development, they're very similar to Bailey's, but we were able to adapt the DAISY-2s more easily to conduct them over Zoom. We are still going to be looking at infants that are 6 to 12 months old, so still very young. And we've expanded a little bit in this paper on the date of birth. So we have infants born between March of 2020 and November of 2021. We also expanded in this paper into Utah and Alabama, so really nice geographic coverage, and have a larger sample size of a total of 407 infants. Unfortunately, here we did not have any pre-pandemic born infants, but we have a nice division between unexposed and exposed infants, and then a small group of infants who we don't know their exposure status, so we consider them a separate group. The really good news about this paper was that we found absolutely no effects of COVID exposure, once again, with this much more expanded assessment. So this unfortunately made much less worldwide news because it was only good news. But this was really a phenomenal finding to have this very thorough assessment show no differences between groups. And finally, I'm going to tell you about a paper that came out just a few months ago, and this is the world's first paper evaluating the possible increased risk in autism screening rates in both pandemic-born infants as well as by COVID status. So here we use both electronic health records as well as our combo cohort with online surveys. We looked at infants between 16 and 30 months of age. The date of births for all the infants that we looked at for the electronic health records was January 2018 to September 2021. So these infants, of course, were born at Columbia University, but also followed up within our clinic system. We had about 2,000 infants total between the electronic health records and our combo cohort. And the really good news here is that we found absolutely no effects of either COVID or the pandemic environment. So this was really good news. Unfortunately, it's very hard to make a good career as a scientist with negative findings, but this was the best negative finding of my career. So I will just end by saying that this, just showing you this infographic of our collaborations. We are happy and thrilled when people want to join us. We've been adding a variety of components to this cohort based on the investigators that come and are interested in working with us. And with that, I will thank all of the many contributors and our funders, and please follow us at Combo Study on X/Twitter, or if you just Google Columbia and Combo will be the first thing that comes up.
Thank you so much, Dr. Dumitriu. What a great way to end our webinar this morning. Four very different and four very interesting presentations. I'm going to drop a note into the chat for everybody because I know that some of you may need to run at 1:30, but if you do have questions for our speakers that we're not going to get to today, don't worry. Please send us an email at info@covidinfocommons.net. It'll go directly to me and I'll be happy to put you in touch with the PI. But I want to also hand it over to Florence for maybe some final thoughts or summary ideas. I can see your eyes moving in the background, which means that ideas are pinging around. So I'm going to.
So easy to read me. Thank you, Lauren. So, well, you all made me think, and I have questions for all of you, but it's the bottom of the hour and we're very sensitive about your time. I'll just do a quick round to share some of the thoughts. Dr. Dumitriu, do you know Rebecca Powell at Mount Sinai? She does work. You do? Okay, good.
I don't know her personally, but yes, I know of her work.
Yeah, because you two could be buddies and you're not that far away from each other. So, you know, she did work on, you know, breast milk and mothers and not separating them at birth because of that. So it's very aligned. And I actually want to share my screen real quickly because this also relates to something that I did. Let me see if I can do this and share the right parts of my screen, because I wanted to mention for Dr. Pollard. So in the COVID Information Commons, we have that, you know, machine learning clustering tool. And so what I did while you were talking, Dr. Dumitriu, is I went in and said baby, mother, put a couple of things in there. And so look at you and Rebecca, how compatible. So the clustering is there. And then what you can do with this is that you can just click on Rebecca. So what's she up to? Oh, comprehensive reactive antibodies in human milk determine their potential as COVID-19 therapeutic and to prevent infection. And her research shows that the babies don't get infected by having the mother's breast milk, which is really important. So it's just very complimentary. I was like trying to read all the little humans in your in your citation. I saw Rebecca, but I think it was a different one. So yeah, that's just a thought, maybe to reach out to her. She has a lot of very cool information. And then Dr. Pollard, I did the same thing for your stuff. Let me see if I can find it now. Uh-oh, you know, when like you share too much, have you seen that ad on TV? So so I actually found some interesting things for yours as well. So I was just going to say, if people want to leverage that tool, it's free and open and available all the time. I also, you know, when you were talking, Gabriella, Dr. Wilson, about you know, grip, you know, I have family in there, and that's the grip, right? And it sounds like that's what they call it when you get sick. And in France, it's very similar. Did they call it that before the COVID vaccine was available, or after, or both?
Before.
You see, that's very interesting because I have a Hispanic friend who lives in Texas, and he kept on saying that it's like the flu. And I'm wondering if there was some thinking that, well, until we get a COVID vaccine, just go get the flu vaccine. I wonder if they thought that would help them. Do you think that was part of it or no?
No.
Okay. Oh, well, I'm trying to give them the benefit of the doubt.
And it was an interesting conversation with the public health department about that. Like, why should we tell them it's different? We don't want them to panic. Well, one thing is not to panic, but another thing is to have the right information.
Totally agree with you. And then, oh, I think Emily had to leave on time. Oh, not Emily. I mean, yeah. Oh, we have two Emilys. Yeah, it was Emily. The other Emily had to leave on time. And so, well, this has been wonderful. I always learn so much, and you make me think, which is my favorite thing to do. So that's all I really had, Lauren, trying to help people find each other so that they can collaborate together. And Dr. Pollard, I'll see if I can find the other one that I created for you before.
Thank you, Florence. Yes, it's been a really interesting set of presentations. I'm so excited to add these forward to our ever-growing collection of presentations on the COVID Information Commons website. I shared in the chat one of the presentations that we were just talking about from Rebecca Powell, so you can see what it is that we do with these talks and how we share them with the community following these events. But all I will say to wrap it up is thank you so much, everyone, for joining us this afternoon. We're so pleased to have heard your research, and we hope that you come to our next upcoming webinar, which will take place in 2025 at some point. Thank you all very much, and have a great rest of your week.
Thank you. Thank you for having us.
Thank you.
Wonderful.
Bye.
Everyone be well and happy holidays.